Hi, everybody out there in the web space. This is Alec McKenzie, co-chair of the Science Committee for FightSMA here with our annual webinar. And we've got a distinguished, attractive panel here today with us. We thought we'd start off, we've got several questions in the queue, but we thought we'd start off with just having the panel, perhaps starting with you, Chris, and working across. Introduce yourself and ask and tell us how you got involved with FightSMA.
So my name's Chris Lorson. I'm at the University of Missouri, and I've been working on SMA for about a long time, I think, about 15 years now, 16 years. And I was introduced to FightSMA back when the meeting was held in Richmond, Virginia in 1998. And Joe and Martha invited me down when I was a postdoctoral fellow working for Dr. Elliot Androphy at the time at Tufts University in Boston. And it was a different style of meeting. It was a small meeting. It was a group of individuals who were really focused on developing, new ideas for the field and really trying to push things forward. And at the time, it was very biochemical. There was no talk about medicine, translational compounds, those kinds of things. It was very biochemical, very genetic, and I think watching it over the last 15 years, it's evolved dramatically. So it's been an exciting ride to witness this. And since '98, I've been coming to most of the meetings as it's moved from Richmond to DC and incorporated different elements, including the government relations arm as well. So it's been an exciting ride.
I'm Elliot Androphy. I'm from Indiana University School of Medicine, and I've been involved in the SMA field for a little bit longer than Dr. Lorson. I was lucky enough to recruit Dr. Lorson to the field of SMA probably about 15, 16 years ago.
1997, July.
Exactly.
Perhaps one of your greatest achievements in SMA.
It is actually one of the ones I'm very proud of. SMA, we got into SMA by serendipity, having cloned the gene without knowing it was the gene, and then saying this is, after it was postulated to be the causative gene, starting to work on it and being honored enough to attract very bright people into the field. I met first FightSMA with meeting Martha and Joe Slay at a NIH meeting, and was particularly enticed to come into the field and stay in the field because of their energy and the group that they put together of dedicated parents who are willing to do the hard work that we could go back to our labs and be able to find a treatment for this disease. And over the past several years, amazing progress has been made. This is a terrific small meeting where we can interact, and scientists can interact with each other and collaborate and advance our progress.
Dr. Craner, what's your story?
I'm Adrian Craner. I'm at Cold Spring Harbor Lab. I've been working on RNA splicing for 30 years since I was a graduate student. So I think it was in '99 that I went to a meeting at the NIH that was mainly based on a paper that was soon to be published by Chris and Elliot and their colleagues that linked defect in splicing of the SMN gene. And so that's how I became aware of SMA and was enticed to start working on it within a year or two. And the way in practice how that happened is that FightSMA gave us a grant to start a project to begin to study why is there exon skipping in SMN2, and then so we continued working on these aspects and got more into drug development eventually. So I've been trying to come to this meeting nearly every year. I've greatly enjoyed the give and take at these meetings.
And it has to be said that, Adrian, one of the compounds that you've been working on for the last decade or so is one of the really true shining stars in the SMA pipeline. So it's been very refreshing to see you work it all the way from the bench top, coming up, working with ISIS and moving it along. Dr. Sumner.
So my name's Charlotte Sumner. I'm at Johns Hopkins School of Medicine. I'm a neurologist and a researcher in the SMA field. I've been working in SMA for about a dozen years, so a little less long than some of my colleagues here. But I first came to a FightSMA meeting not long after I got into the field, and I was fortunate. I was working in Kenneth Fishbeck's lab at the time, who was at the NIH, which is nearby, and he was invited to the meeting, and he was allowed to bring me along. And it was in part coming to FightSMA meetings that really got me excited about this problem and working on this problem for my career. And I think it speaks testament greatly to Joe and Martha that they've created such an atmosphere that's so open and collaborative and fun to really talk through these ideas. And obviously we've made a huge amount of progress, so I'm grateful to them.
Great. Brian.
Brian Kaspar from Nationwide Children's Hospital and Ohio State University. I've been recruited into the SMA space with, real pioneers within this disease, from Arthur Burgess, Alex McKenzie, Chris Lorson, and Martha Slay. I think it was back in 2007 where I had started a laboratory, and they recruited me. That list of that group brought me in to start learning about SMA, really in the formative years of my laboratory in becoming independent on doing research. And we've always had a notion to try to develop drugs and or therapies that could target the brain and spinal cord. And really, it was this group that really brought me into the field to learn about spinal muscular atrophy, and it's a privilege to be here today. Thank you.
We're privileged to have you, John.
So I'm John Porter. I'm a program director at the National Institute of Neurological Disorders and Stroke, which is one of the 25 components of the National Institutes of Health. So what I do, I'm responsible for a number of neuromuscular diseases, including SMA. And so what all that means, basically, is that I try to facilitate funding for SMA. And so I work with the other folks at this table, as well as the other investigators at the FightSMA meeting, to try to help them get funded to do research on the disease. And I'm very interested in forming partnerships and with advocacy groups, with companies, so that the government funding can help leverage other funding and move this field toward a therapy.
And we have to say John is stepping away shortly from 10 years of hard work at this space within IDS, and we have a debt of gratitude for all the work that you've done for SMA and the other neuromuscular disorders, John. Thank you very much.
Alex.
Okay, enough getting all emo here. First question, and it may not be the first time, Brian, last time that you're asked. It's from Italy, Rome, Italy. It's Maria Riccardi. Good evening. I'm writing from Rome. I'm a mother of a 13-year-old boy affected from SMA2. Maria wants to thank us all for the work, all you for the work you do, and the commitment that you put in every day for our children. My question is for Dr. Kaspar. Is this, once you've passed phase 1, the gene therapy, shall it also be accessible for patients coming from foreigners, I guess coming from foreign lands? Thanks and good job. Maybe just to interject here, perhaps you could just give us a sense of where you are with the gene therapy timeline right now, and a sense of what the next steps are.
That's great. Thank you, Maria, for your question, and also from writing from one of the most beautiful places in the world, Rome. Can't wait to get back and visit. It's a pleasure to give you an update, because I know that there's many people who are going to be asking the question, where do we stand with our gene therapeutics, our AAV9, to move to human clinical trials. And what I can say is we're extremely close at the moment. There's been 2 first-in-human trials, and that is starting a phase 1/2 FDA-sanctioned human clinical trial in type 1 SMA patients, type 1 patients with 2 copies of SMN2 that are 9 months and younger. For the first-in-human trial, there's going to be 9 patients in the first trial, and we have product produced So our GMP-produced virus is ready to enter human clinical trials. There's final checks and balances going on this. There's a number of regulatory bodies that we're dealing with still, and we're on the final yard line, is what I'd like to say. We're expecting at Nationwide Children's to release a date in the very near future. So we're extremely close to being able to enter into phase 1/2 clinical trials in patients. So please be looking on clinicaltrials.gov for additional information, and I'm certain that Nationwide Children's will be releasing more specific dates in the very near future. What I can say is things are going extremely well, and we're very close to the initiation of these trials. Dr. Jerry Mandel at Nationwide Children's will be running the trial in Columbus, Ohio. Your question is extremely good. You mentioned your son, was it son or daughter?
It was his son, I believe.
Yeah, your son has SMA type 2. We are not forgetting about type 2 or type 3 or even older and larger type 1 patients. This remains a critical focus to our research-based studies and towards our future human clinical trials. Our goal is to run a phase 1 trial in the phase 1/2 trial in the type 1 patients with perfection, and that is running a trial where we can demonstrate safety and where we can hopefully demonstrate efficacy in SMA patients. It is our critical goal to do our clinical trials with perfection, and that is enrolling the right patients for the first in human trials. And Dr. Jerry Mandel is doing a fabulous job with the team that includes a battery of individuals at Nationwide Children's and throughout the United States and world that we're collecting information on from a clinical perspective. We have lofty goals on trying to think about developing this therapy towards type 2 patients. Clearly, with success in a first-in-human trial, we would rapidly push towards entering into older type 1 patients, type 2, and type 3 patients. I think we have a lot of exciting preclinical data to suggest to date that we can actually do something good in type 2 and type 3 patients, and we're excited about moving all of these therapies forward for all SMA patients.
Great. Thanks very much, Brian, for that very thorough answer. This next one is from Nicole Lamonaco Soon from Long Beach, California, and I'm not sure who would be best to handle it. Nicole is an ambulatory type 3, have lived with this condition now for 30 years. Very thankful that soon I won't have to anymore. I guess whoever answers it can touch on that. With that being said, when will adults like me be able to benefit from and be part of trials like ISIS? We are aware, so far the adult community feels a little left out. We are aware of very few trials that are available, but none really for us. We are once children too and have children who would love to see their moms be able to do more, thankful and grateful and ready to receive a treatment. So a bit of a tricky one. I don't know. Charlotte, would you like to give that a whirl?
Sure. I mean, I can certainly comment. So Nicole, it was?
Nicole, that's
yes,
right.
Nicole. So it turns out that I take care of a lot of adults with SMA, so I really understand your perspective. This is a common feeling with adults living with SMA. That
You're an adult neurologist.
I am an adult neurologist,
Yeah, right.
and so I understand very much what she's feeling. There is a sense that that adults with SMA are not receiving the same attention as children with SMA. But I think that's a bit of a misperception. I don't think that we don't believe, first of all, that many of these treatments that are initially being tried in children with SMA will be very effective in adults with SMA as well. So that's one very important point. I think particularly we're in the past, we've particularly thought about doing clinical trials, for example, in ambulant adults with SMA. So valproic acid, which many know was tried in various types of clinical trials, there was a clinical trial specifically devoted to adults, ambulant adults with SMA. Unfortunately, that trial didn't show efficacy. That was a trial of valproic acid that was done by John Kissel at OSU. But certainly there is attention towards thinking about how to do trials specifically in adults with SMA. And I personally believe that these emerging treatments are going to be very effective in adults as they are in children. The adult community is a smaller community of patients, and I think that that's the principal reason why many of these therapies are first being tried in children in these clinical trials.
Okay, thanks, Charlotte. And Adrian, I mean, I don't know if you're au courant with ISIS and its plans as far as phase 3.
I mean, my understanding is that the criteria that went into selecting which patient population to first try the drug on. I mean, so part of it is what is the fastest way and safest way to show both safety of a compound and efficacy, and so that takes it's a sequence of events that have to happen, and so initially it was for type 2 and 3 patients, age 2 to 14, then 2 to 15, but now there's a separate trial on infants, much younger infants with type 1 SMA.
Right.
I mean, if the trials are successful and the drug is approved, or possibly before that, I imagine that these trials will be expanded to include broader populations of SMA patients.
Good. Chris?
Just to comment on that, I think that as well, all of the data in the preclinical models suggests that the earlier the better. And so in terms of a patient population, to prove whether or not there is an effective reaction within these various therapeutics, we want to give every drug the best possible opportunity to prove itself, efficacious. And choosing the right population in terms of proving a yes or no answer might mean that it is an earlier population. But I don't think anybody is under any illusion that an older population is not as important. Excuse me.
And we heard at the meeting some encouraging research showing that certainly in mouse models that basically even with established SMA, it did appear to be some beneficial impact of some of these therapies. So.
Right. We saw it in several animal models demonstrating that even post-symptomatic, that there's a significant benefit in terms of the SMA phenotype.
I think there's also data in preclinical models in mice that the same drug delivered to a severe model versus a less severe model, the efficacy can actually be greater in a less severe model. So work from Christine DiDonato with the drug we've studied showed that.
Yeah. Chris, we'll stick with you for a second. Brittany Roberts, a fellow Canadian at Pointe de Chêne, says, out of all the research being done, is any of it working? How long do you estimate it will be until we have a cure? And so we're just coming off our two-day meeting, and Chris, I don't know if you want to just hit some of the highlights with that regard.
I think some of the things that we hit upon in the meeting really demonstrate that this is not, to go Canadian on you, this is not a single shot on goal. There are multiple shots on goal. And I think that many rare diseases would be incredibly envious in the sense that we have a bona fide target. SMN2 is this amazing molecular rheostat that's waiting to be turned up. And I think that's what you see with small molecules. I think that's what you see with antisense oligonucleotides. That target engagement, you can demonstrate that in cells, you can demonstrate that in animals, and moving that into the clinic provides this great platform to move these kinds of compounds forward. Now, we know what the gene is. It's SMN. So gene delivery, restoring SMN expression is obviously kind of, the ultimate goal. And that's what the AAV vector could be. So I've talked about gene delivery, I've talked about antisense oligonucleotides, small molecules, as well as kind of a broader perspective. There's neuroprotectants, and this was just a compound that was approved in Europe or has finished phase 3 trials in Europe, a Trophos compound. So this is a neuroprotectant designed to kind of create the appropriate milieu within the CNS for motor neuron survival. So I think that in terms of SMA moving forward in a therapeutics perspective, there's an amazing amount of work that doesn't necessarily overlap, but takes different approaches to the same end game. And I think that's incredibly powerful, that it can demonstrate target engagement as well as preclinical efficacy. And I think that really puts SMA forward in terms of rare diseases, and it provides a considerable amount of hope, I believe.
John, what are your thoughts? You get to scan the horizon fairly comprehensively from Duchenne to SMA to other disorders. Are SMA doing well?
So I think there's a lot of opportunities in SMA. So the NIH, actually, we're fortunate to fund everybody at the table.
Yeah, we chose them that way.
And lots of other people that have been at the meeting the last couple of days. So, you know, what I can say about this community is it's a very collaborative group. So meetings like this, small meetings, lots of interaction. There's lots of data sharing, lots of resource sharing among the investigators in this room, and that's really what it takes. I mean, certainly research is competitive. Our funding, we fund less than 20% of the grant applications we receive, and so everybody that gets a grant application is already upper crust as far as a researcher goes. So do what we fund for SMA, is it enough money? Certainly not. And that's a function of our budget, and it's a function of the competition for grants. But I think when you start with a community that is united as this community is, that is partnered as they are, and shares information closely, and the level of expertise I see in the companies that have been attracted to work in this field is huge. I mean, I think it's very much the envy of other diseases that my institute funds, to have companies that really go after understanding the basic disease mechanisms and then trying to exploit those disease mechanisms to develop candidate therapeutics is really tremendous. So I think this community is really in great shape. Can we do better? Of course we can. But I've really been impressed by the last day and a half, 2 days at the FightSMA meeting about the level of collaboration among the investigators here.
Yeah. Okay. Well, that's it does strike one as a good sociology that one sees in the room. Yeah, and yeah, it's an enjoyable one as well, as Charlotte says. Here's one from Michelle W. asking about, well, what is the role the mitochondria, the powerhouse for our cells, plays in SMA? Elliot, perhaps you're a good basic scientist who's doing great basic work and who understands the mechanism as well as anybody.
Well, I think that's the role of the mitochondria is in energy metabolism for the cell, and we're just beginning to understand how the low levels of SMN infect intracellular processes, which is, we know the target, as we've all said. We all know that increasing SMN2 expression of the SMN protein will restore its function. But the underlying question is, if we knew what the functions are that were essential for motor neuron development and sustenance, that might also contribute and give us new avenues for treatment. So as you mentioned, mitochondria may be something that are involved in the pathogenesis, the underlying reason why the weakness develops, those motor neurons become dysfunctional. And so that's something that has been explored. There are processes in the cell that can use these organelles, these things like mitochondria, for maintaining the cell, and in my lab and others are interested in understanding what happens to mitochondria in other parts of the cell when the cell is stressed, and that may be important for maintaining and survival of these cells. So I think that's beginning to be looked at.
Do you have a sentence or two on COP1 that you can give the general public that is comprehensible?
Yeah, well, comprehensible.
Sorry about that last edition.
So what Dr. McKenzie is referring to is my lab has been interested in something we call a vesicle, which is a transport complex, and it moves things around in the cell. And we've shown, and other labs have followed and shown this too, that this interacts with the SMN protein. So as you probably know, the SMN protein moves around. It's made in the body of the cell and then can move into the distal parts of the neuron and the axon. And it's doing that for a reason, and it's probably transporting cargoes or goodies for maintenance of the cells. So we're interested in how this transport vesicle is involved. And vesicles, it's like an organelle, like the mitochondria that you're talking about, how they interact functionally with SMN.
Great, thanks a lot. I get it now. We're said that nutrition and ventilation play an important part in SMA. This is a tough one. I don't know, Charlotte. What is being done to look into, say, the AA diet for type 1 SMA patients? That probably isn't your table necessarily.
No, I'm not an expert in that area.
Sure.
I think there are others trying to investigate
Yeah.
issues of nutrition in SMA kids, but it's a very difficult problem and a difficult one to study.
Yeah, I would. Yeah. I think there's a lot of adherence to simplified diets, especially in the type 1 community, is not, to my knowledge, a body of evidence that necessarily supports it, but those studies haven't been done, and that's perhaps something that we should be looking at. And then going out of the box, has anyone looked into the use of essential oils, a company called doTERRA, to help symptoms of SMA or activate the SMN2 gene? Does that ring a bell with anybody? Not really. Okay. But by and large, we look for a body of peer review evidence behind any therapies that are offered. And so there are a lot of therapies out there which are probably not as what they appear to be. And this possibly is one of them.
I mean, I would just say to any family considering trying a specific diet or trying a specific vitamin or nutrient to really supplement, thank you, Adrian, to really talk with your physicians carefully about it. Right. Many of them won't be harmful, but some are.
Right.
And so it's really important to talk about it carefully with your physician.
Stacy Edwards from Independence, Missouri, asked, do a spinal fusion rod affect the ability of a child to be treated with gene therapy? Now, Brian, I don't know if that's, or Charlotte, whether you have any thoughts about that.
So I think Brian will comment on this, but I know for the initial ISIS trials, because the drug is being given intrathecally, having had prior spinal fusion surgery is an exclusion criteria for the initial trials. And the thinking here is that it could complicate the delivery of the drug. But, that will certainly change going forward. I don't know what, in the gene therapy trial, what's being planned.
Currently, our plans with the type 1 patient population is so young for the first enrolled in human clinical trials, 0 to 9 months of age, that we don't have, that's not even on the radar of a 9-month-old needing any sort of rod placed for this purpose. However, what I think we would say is, as we move forward, there's nothing to think about a procedure like that would preclude you from being in future clinical trials. I think in the first in-human trials, particularly with gene therapy, are now being planned for the youngest of patients. As we move forward in the field, we will certainly be working with the Food and Drug Administration as we expand human clinical trials into older patients who may have other sorts of surgeries being performed on them. And this is something that we'll be working very closely with the Food and Drug Administration on. There's certainly no reason to think that a rod would preclude anyone from any future clinical trial, be it gene therapy or any other drug sort of therapy. These are things that have to be, though, broached with the Food and Drug Administration and the regulatory bodies that move forward, that really make the decisions of first-in-human sorts of trials. And it's up to us to be able to provide the rationale and keeping safety at the primary of importance. So once again, I don't think there's not many things that would be out there with a successful therapy that is proven safe in first-in-human trials. There's not too many things that would preclude individuals from being in a clinical trial. Again, that's for the regulatory bodies to really have the final say on. And I think this is, with success, the doors will certainly open towards all patients with any of these therapies, including gene therapy, that's moving forward.
Great. Thanks very much for that. I have to say that there are more questions coming across, and I think we'll have time to answer to the people who are listening closely out in cyberspace. And some of the more orthopedic ones, for example, do not fall necessarily into the wheelhouse of our panel. So for those questions that we don't have time to answer, we'll make every effort to get them to a person of individual of expertise and get an email response to them if we don't get time to get to the actual question real time. There's one, Charlotte, this is you again. It's quite a scientific one. This is from Fred in Ohio. Is the work of Martinez Hernandez, synaptic defects in type 1 SMA regarding acetylcholine vesicle distribution and packaging at the motor nerve terminal suggested of a systemic anomaly of acetylcholine everywhere in the autonomic nervous system or just in the motor neuron itself? So that's clearly, I think, from a scientist, but can you do your best with that, Charlotte?
Good question, Fred.
You want a job?
I mean, this is something we've talked a lot about at this meeting over the last couple of days, and I'm sure everybody on this panel could talk about this issue. I mean, I think the fundamental, well, maybe there are two fundamental questions in Fred's comments. One, is this disease of SMA purely a disease of motor neurons? That is, this ubiquitously expressed protein, SMN, is present, we think, in all cells. And yet we've traditionally conceptualized SMA as being a disease specifically and only of motor neurons. But there are experiments that might suggest that, in fact, other cell types, other tissues may play an important role in this disease. And in the scientific community, we all spend a lot of time wrestling with this and trying to sort this out. It's not only very important for us to really understand the best basic mechanism of the disease, but also, of course, has major therapeutic implications in terms of where do we need to restore SMN. Is it just to motor neurons or is it to other cell types? And if it is to other cell types, what are those cell types? We, at this meeting, I think heard some data that would suggest that it is principally motor neurons, but other cell types may contribute. Exactly what those are, though, we're really, I think, still unclear about. The other part of Fred's question that's embedded in that is, if we accept that the principal problem is in motor neurons, which, I think all of us would agree that the principal problem is there, where in the motor neuron does the problem start? Is it in the cell body of the neuron, which sits in the spinal cord? Is it the wire of the neuron, the so-called axon, or is it in its connection with the muscle, what we call the neuromuscular junction? And there, too, we wrestle with this issue in the research community. There is data to suggest that this connection part is something that goes early, but there's other data to suggest that it's a more widespread abnormality within the entire motor neuron. So very good questions that I think all of us are actively trying to sort out.
Okay, good answer. Question from, does anybody else have? No, okay. You did it. Katie asks, I have saved all blood core, I've saved stem cells from all three sons. My youngest son, who has SMA, is 3 years old. Could my youngest benefit by using stem cells from his brother for gene therapy, which is currently in phase 1? I don't know if anybody wants, okay. Are you handing off to Dr. Porter or Dr. Casper there, Charlotte?
I was thinking about Brian.
Okay, yeah. I mean, it's the third rail of where, I mean, it's two combined in one. It's asking about the gene therapy, but it's asking about stem cells.
So, Katie, you're asking a complicated question here, and what we know in the field on stem cells, and I'm not aware of exactly the stem cells that you have banked, but let me just give a more general overview of where we stand in the SMA field to date with stem cell-based therapies. And please, anyone on the panel, correct me if you disagree with this, but I think where we sit on stem cell and the stem cell transplantations that have happened in preclinical, so this is mouse data only, is that many types of either neural stem cells, mesenchymal stem cells, and even motor neurons generated from a stem cell population has been transplanted into the mouse model of spinal muscular atrophy, so the Delta 7 model that Dr. Arthur Burgess developed at Ohio State. And the results on transplanting stem cells into this mouse model have been modest. So what we've seen is short extensions of survival, unless anyone knows of something that they could say that we should be extremely excited about. I think it's early days with stem cell research for transplantation into the spinal cord. That does not mean to say that Brian Kaspar is against stem cells. So at the present state, what individuals, and there's some very good laboratories working on stem cell models, and this ranges from things that are happening in the United States to things that are happening in Italy, as far as basic research studies that are showing neurotrophic effects of transplanting these cells into the spinal cord that are then protective towards motor neurons. But again, the results in the mouse model of the disease have been modest. What I would say is there's a lot of potential in stem cells for stem cell regeneration and being a little bit more specific, stem cell replacement, motor neuron replacement. Actually, anyone who says that they can replace a motor neuron, I will be as bold to state they are lying to you. You cannot replace a motor neuron at present state with the science that we have today. You can put a motor neuron into the spinal cord, but there has to be a lot of things that happen to call it motor neuron replacement, and that is the motor neurons that you transplant into the spinal cord have to be properly connected, and then they have to go out and escape the spinal cord and make a new connection to muscle, to the right muscles, and start functioning properly. There's a lot of things that have to happen for that to be successful. I'm not saying that it's science fiction to date, today, but we have a lot of research to do, and I think this is, I think everyone on the panel would say, in spinal muscular atrophy where we lose motor neurons, the penultimate therapy would be to replace motor neurons. Unfortunately, science isn't there right now. And there's a lot of work, and there has to be a lot of basic research that needs to go on to make this a reality. It's not going to happen overnight. There's many groups, including our own, that have this science fiction. It's not science fiction. There has to be a lot of basic research, a lot of understanding to make this a reality. So I think there's hope for stem cells and for the penultimate stem cell replacement. But currently, right now, I think we should be excited about many of the therapies, from oligonucleotides to drugs to the gene therapy, that is actually hitting the disease target, and that is trying to upregulate SMN expression. And those are shots that are much more on goal than to think about a motor neuron replacement right now. But ultimately, there's a lot of research that needs to be done in the stem cell replacement field to try to make this a reality, because ultimately that is the fix. In SMA, you will lose motor neurons, and we know in the field that you have non-functioning motor neurons and or death of motor neurons. Mm-hmm. And the only way to fix that is to replace those motor neurons. A lot of work still to be done. I think we have hope, though, that science will continue to move.
Just thoughts on any stem cell therapies offered outside of North?
Alec?
Beg your pardon?
Can I interrupt you?
Absolutely.
Thank you.
That's once for Chris. And okay.
All I was going to say is that I think if you take a step back and look at gene therapy, even, if you go back 30 years, gene therapy in its infancy, relatively speaking, had so many hiccups. Yes. And I don't feel that stem cell biology, in terms of a translational or therapeutic wave, has hit that. And gene therapy has had and I don't mean to say so many failures, but it has been on its deathbed once or twice. Mm-hmm. And only due to amazing science and really dedication by people like Brian and others that it's been resurrected. And if you look beyond the SMA space, I think gene therapy is really looking at a resurrection of sorts in the last 3 to 4 years, as not just a biological tool, but really a translational tool. And it's because science has been done appropriately, carefully, cautiously, and moved forward in that kind of respect. And I don't think anybody at this table would say that stem cell work and stem cell potential is anything but exciting. I mean, I think that's fairly obvious. But like Brian said, motor neuron replacement might be a long ways away. But if you look at other fields, such as cancer biology, in terms of an ex vivo application, that is moving forward incredibly rapidly and showing amazing success. So I think it just has to come in different spurts and waves. And in terms, I don't think you want to throw anything out. I just think that there's a lot more work to be done in terms of a transplantation and regrowth of motor neurons. I think that bar is a lot higher than, like Dr. Kaspar suggested, some kind of neurotrophic effect that a stem cell therapy might actually elicit.
Charlotte.
I do think this is a very confusing area for patients and families, because you certainly can travel just to Mexico or other parts of the world and be offered lots of stem cell treatments.
And there's very compelling testimonials on the web, et cetera, for these places.
It's a really hard hook not to bite onto. I mean, it's very confusing, I think. And I have a lot of patients who have sought out such approaches in other parts of the world. And I would just say, I think Brian's strong message is reasonable in that there is no evidence currently that's going to that receiving these therapies at this time will be beneficial, and there is real potential
for risk
for toxicities and side effects that could be serious, particularly when injecting them into the nervous system.
So these patients of yours who do go, are you seeing a marked benefit in any of them who go to the Mexico or wherever?
No.
No. I think that's the hard answer. Yeah. Okay. Maybe, Chris, this is back to you. This is Mr. Koulos, who sent about 8 or 9 questions. One of them is the timeline of when, and he mentions isotrophus PTC, gene therapy, when we can expect to see some of these things dropping out. I mean, maybe just a brief going over of those 4 modalities might be useful, and then a sense of the timeline of how it's going to go. And Adrian and Brian can chime in.
Sure, as there are probably other members on the panel who could address this as well. I'll just throw a general one out there. With the ASO work is ongoing and has multiple trials ongoing. And I believe that they're trying to spread into a type 1 trial right now. Is that correct, Adrian?
Yeah, that's ongoing.
And that's ongoing. And sorry, you should probably answer that one. But for PTC, that has been ongoing. They announced that a couple of months ago. That's in collaboration with Roche. That's been ongoing in Europe. And so that's a phase 1. And I believe that's concluding fairly shortly.
Right.
Sorry?
Charlotte, do you...
And we've talked about the gene therapy, and Dr. Kaspar has said that's imminent.
Yes.
Weeks, months. And what was the other one? And Trophos has wrapped up phase 3, and I believe is waiting for phase 3. Phase 2?
Phase 2.
Okay.
Yeah.
So really, I think everything is moving forward immediately.
Right.
In terms of approval, that's obviously going to be addressed by regulatory bodies.
Right. I think there's a reasonable expectation that within the next 2 years, we'll probably be seeing approval of at least one of these, it has to be said, very exciting interventions. The evidence we saw over the last 2 days was, certainly for 3 of them, looked very, very encouraging. I don't know, Adrian, did you want to add anything from the ISIS?
No, that was about right, yeah. So the ISIS trials have been ongoing. At this stage, it's mainly for safety and beginning to look at potential efficacy for a couple of years, and everything seems to be going well, and some of the data have been presented already, and so one could imagine if things go well that 1 or 2 years there's potential, if everything goes well, to seek approval.
John, you've been a huge enabler of all this. Is that your sense as well?
Yes, I'd agree. I think there's a lot of careful efforts. Somebody mentioned shots on goal. It's really important that we have multiple efforts directed in this case. It's the great thing about SMA, and there's consensus on a target on upregulating SMA in one way or another. And I think we've got a lot of carefully planned efforts from some very knowledgeable folks, a panel at this table, as well as the companies that are involved.
So that's the future. Meanwhile, they're asking, Mr. Koulis again is asking, are there ways of addressing SMA right now? Physiotherapy, occupational therapy, vitamins, supplements, amino acid diet, range of motion, other interventions. I don't know, Charlotte, we seem to be picking on you. What's your sense? So if we're not getting gene therapy today, we're not getting ISIS today, what do you tell patients as far as changing the natural history of their condition?
Well, I think that all of the things that Mr. Koulis mentioned, with the exception of not knowing exactly the right diet, is really important. And I spend a lot of time when I care for my patients with SMA talking about how best to address a whole range of issues. They vary depending on the severity of the disease and what SMA type we're talking about. But certainly, major issues of respiratory support and care, nutritional support and care, musculoskeletal support and care. So all of those things are very, very important. I don't have any data to back this statement up, but I do believe that maintaining all those factors optimally improves outcomes.
Right.
And I'm also, in older patients, a big fan of regular exercise programs. I think they also are very important. So I think it's very important that all individuals with SMA have a physician who understands their disease and can work with them to optimize their health and quality of life. And there is a lot to be done in that regard.
Right.
Charlotte, so if I can ask a question. So for the families out there that don't have the benefit of a physician in a major academic medical center like yourself that really knows the disease, so care standards have been put in place for SMA, correct? That to help guide, quote, the ordinary physician out there that doesn't have the advantages that you do, to try to assure that there's a good universal standard of management for patients, right?
Yeah, John, it's a very important point. So this, a lot of effort went into developing those standards of care. So a number of experts from around the world got together, spent a huge amount of time coming to consensus about the optimal way to help individuals with this disease. And it is a document that obviously all physicians can reference and use as a guide.
So the families need to make sure that they access that document and that they're pushing their personal physicians to get that standard of care. Don't assume that that it's going to be there, but be proactive and get a hold of those standards and push your personal physician.
John, while we have you, this is a Dr. Anna Caspi from Life Technologies brand at Thermo Fisher Scientific. He's saying, she, I apologize, we're in a very interesting regulatory time. FDA is working under a very antiquated pharmaceutical model that for novel therapies that are so target-specific, like gene therapies and antisense, the notions of efficacy that FDA espouses do not really match the targeted therapeutics. I'm not quite sure I could catch that drift. Meaning a patient that is responding to therapy may have increased expression of SMN2, but won't start walking with X days of drug administration. What is being done on the regulatory side to educate the FDA so the trial aims for efficacy are matching the therapies?
So first, I try not to comment on the FDA.
Yeah.
So the NIH is responsible for funding research in these disorders, and that's a very different mission from regulatory. But I will say
Speaking of hockey, that's very good stick handling.
Yeah, I will say that the FDA has launched a number of initiatives to start listening to the patient population more. And I think there's increased opportunity for rare disease communities like at the SMA community to access the FDA, to make sure that they understand their diseases, to make sure that they understand what the patients regard as important in goals for therapies. And I think FDA's clearly got that message, and it's a huge part of the regulatory approval that the endpoints of clinical trials designed by the group of folks at this table and then the companies out there take to heart that patient voice and what they consider important, what the parents and as well as the patients consider important.
Okay, good. Well done. Chris, this is from Casper in Europe, perhaps a distant cousin, Brian. 3 years ago, the team of Dr. Lawson, and actually this is for Chris and Adrian, the team of Dr. Lawson reported that ASOs were incomparably more effective in SMA mice when administered systemically rather than ICV. Okay, this is more Adrian. Why are the current clinical trials of ASO, in particular ISIS, employing the ICV route instead of using systemic delivery? Is there work going on to use systemic delivery of ASOs? So that sounds like it might have been your work that was being referred to.
Yeah, that was addressed during the conference or discussed. There's still work ongoing in that area. So it is true that in the preclinical work in mouse models, our work in collaboration with ISIS showed a surprisingly strong effect of administering the antisense oligonucleotides systemically. There was also a benefit of administering it directly to the CNS. But so that was a bit unexpected. We're trying to understand what it means. One interpretation, though, is that this may be a feature that's characteristic of the mouse models, the very severe mouse models, and also the fact that without treatment, these mice only survive 10 days or 15 days, and you have to do the treatment very rapidly after birth. So there's certain features of a newborn mouse, and so it's not clear, how much of that applies to a human patient. So ultimately, I think that that one needs to explore the possibility of delivering the drug systemically or both systemically and as it is being given now intrathecally. That's my opinion, but that requires appropriate safety studies to be done for delivering systemically before it can be done in the clinic. So that'll take additional work and time. I'm not sure when it's going to be done. But ultimately, I think it needs to be tested in clinical trials whether there's an additional benefit and to what extent by giving the drug in different ways.
Dr. Lawson.
And I think this also gets back to what Dr. Sumner was speaking of earlier, and that I think that there are several questions out there still, but one of them happens to be what tissues need this induction of SMN. And so I think that there's still room for debate, and delivery route will impact what tissues see the drug. And obviously, a more CNS type of approach favors the CNS, more peripheral should favor peripheral, but depending upon the drug, you might get both.
Right.
But I think that gets back to the fact that there are still biological questions that we need to address, and that this impacts how trials and therapies are designed. So I think that's a great question, but it's one that we're wrestling with right now.
Right. And it's certainly one, Brian, you're covering off the systemic versus intrathecal from the gene therapy perspective as well, are you not?
Our gene therapy, we're first moving into clinical trials with a systemic gene delivery, and we're about, don't hold us to the timeline, but we're about a year out on a CSF delivery. We're trying to move rapidly on that program as well. You know, I think there's, when we look at the data in the mouse model, certainly we would agree with Adrian that systemic delivery of a therapy that increases SMN results in the largest extension of survival in these mouse models. How important will that be for the human clinical trials? And I think that we have taken an approach to say that we're open to any scenario, and therefore we need to systematically move forward with the therapy that we have advanced furthest at this point, and that is the systemic delivery program. This may matter more when you're thinking about a type 1 patient where there is an autonomic nervous system problem. And I think there's debate within us researchers of trying to figure out what's wrong with a mouse. But what we do know in most type 1 SMA patients is that there appears to be an autonomic nervous system disorder. And that is, many of the type 1 patients do not regulate temperature very well, hot and cold. There's also many signs of cardiac involvement from bradycardia and other
slow heart rate.
situations that might suggest an autonomic nervous system disorder. And there's also then the digestive, the bowel movement, and the constipation issues that also suggest the enteric nervous system, also autonomic nervous system. I think that we have to just pay attention and say that motor neurons are probably the primary, as I think we all agree, motor neurons are a primary cell that has deficits due to low levels of SMN in this disease. But there are likely other cell types, and I think it's including other neurons, potentially autonomic nervous system connections that could be involved and are likely involved in this disease. So we have to remain open and really allow these therapies to move forward. We are open to a CSF therapy working. We are open towards, we would like any of these therapies to have a clinical, meaningful effect. And I think this is, for any of these programs, it's critical, absolutely critical, to design the first in human trials to, number one, allow safety, and then number two, allow one to catch or document a clinical meaningful effect. And that's what I think we're trying to do as a field, really working together with clinicians. The mouse can get us so far, but then we have to get into our experimental trials.
Yep. The human as an animal model. Charlotte, do HDAC inhibitors work? And on which cells is the question? Perhaps you could just tell us what HDAC inhibitors are first.
So HDAC inhibitors are a group of compounds. They've actually mainly been developed for oncology purposes, for cancer purposes. They're drugs that inhibit a group of enzymes that are called histone deacetylases, and these enzymes modify what are called histone proteins, and histone proteins associate with DNA itself. So DNA is not the simple sort of linear structure that we think of it. It actually wraps itself around histone proteins, and DNA can be relatively tightly coiled or it can be quite open. And when it's more open, genes tend to be expressed more robustly. And an HDAC inhibitor actually works to sort of open up chromatin and allow genes to be expressed. So the idea behind trying an HDAC inhibitor in SMA arose some time ago. It stemmed from observations that if you gave histone deacetylase inhibitors to cells, that would result in increased SMN expression. And then we and others some years ago now showed that if you delivered such drugs to SMA mice, that also improved the phenotype of those mice modestly. We now have numerous other drugs that have shown much more dramatic effects in SMA mice. Histone deacetylase inhibitors were some of the first compounds to show some efficacy, so they were a very important proof of principle that increasing SMN could result in an improvement in the SMA phenotype in SMA mice. But in retrospect, their effects are quite modest relative to some of the other newer therapeutic agents. They also have another downside, which is that they activate a lot of genes. They're not particularly specific for SMN. Many, many genes are changed in their expression with these drugs. And so there are lots of potential toxicities from these drugs, whereas, again, some of the newer agents are much more specific for SMN, and that's what makes them so exciting, in addition to the magnitude of their effects. Some of the HDAC inhibitors were tested in clinical trials in individuals with SMA. The two drugs that were taken to clinical trials, one was called valproic acid, the other phenylbutyrate. These were two HDAC inhibitors that were already in clinical practice. So we had been using them for other purposes, valproic acid for epilepsy and phenylbutyrate for some metabolic diseases. And that's why we were able to take them to clinical trials so quickly. They're weak HDAC inhibitors, and in the clinical trials did not appear to increase SMN expression very much. And in that context, they did not show substantial efficacy in patients. Now, whether in the future an alternative HDAC inhibitor might be efficacious in SMA is possible. But I think some of the current new drugs that we've been talking about are much more promising at this time.
Great answer. Great answer. Dr. Larson, if we could just ask you about the National Pediatric Trial, which Martha Slay and Steve Eichenauer and others at FightSMA have been so instrumental in pushing through. Can you just give us your understanding of what that is and how that might aid move the field forward?
This is a bill that has been working its way through the system, initially perhaps as the SMA Treatment Acceleration Act. And this is something that the SMA community has been hoping for and pushing for the better part of a decade. And it initiated with language in Congress identifying SMA as an important disease and worthy of being mentioned on the floor. And it was an evolution, to say the least. And although a number of people within the FightSMA community had infinitely more to do with it than I did. I happened to be up here. And Martha and Steve Eichenauer and Joe and the rest of the government relations team have really crafted this bill into such a way that the NPRNA is designed to help facilitate clinical trials in pediatric diseases. And in particular, when members of Congress got up and they spoke about the bill, most of them spoke about spinal muscular atrophy. And that really, I think, goes back and harkens back to the years of support that families have put into this. And, you know, there are a number of rare diseases that would like a disease-specific bill, but I think when you have families go onto the Hill year after year and tell their stories and talk about how this disease really impacts their family and how it impacts the community, and it's not just one disease. I think SMA stands out and might represent a number of diseases. It represents other rare diseases. It represents a number of struggles for individuals and communities. So the NPRNA is an opportunity to move forward in terms of providing an infrastructure for clinical trials for rare diseases like SMA. And I think that's the overall goal, is to help SMA move through this point. There's been an enormous amount of preclinical work, and you've heard about it by the people at this table and others that have really brought SMA to the forefront. and it takes an enormous amount of financial support to get something like this through clinical trials. So like Dr. Porter had mentioned, there's an amazing amount of collaboration that goes on, and the collaboration stems from the government to the foundations to just the community at large. And I think coordinating this has been spectacular, and I hope this is the kind of thing that really pushes SMA from, first in line to crossing the line.
Thanks, Chris. Adrian, are you done? The ISIS SMNRX, it's a huge success story. Is there anything else that your lab can do to improve on what that reagent is, or any sort of modifications, or is that point final at the end of a chapter?
No, I mean, just to clarify, so I worked with them for several years in developing this, but I'm not involved in the clinical trials, just to clarify that. And if things continue to go well, the scenario is that Biogen Pharmaceuticals will commercialize the drug if trials ultimately prove to be successful.
Early indications are promising, we have to say.
I think things are looking good for what's been presented so far, but there's much work that remains to be done to demonstrate efficacy. We continue to work on understanding. We understand how the drug works, but in terms of where and when these types of issues that in our lab we continue to try to address using mouse models. But there's always potential for improving. There's down the line perhaps greater potency, newer chemistries. And, so we continue on that basis. We're also interested in other rare diseases or other indications where splicing is involved. And in that sense, this was the first work that we did for SMA, so that helps us think about other
familial dysautonomia and other disorders like that where there's splicing involved. Mm-hmm. Here's a bit of a metaphysical question, Elliot. Is it possible that new breakthroughs would come from non-SMA labs? We seem to give you all the hard questions.
I think there's always the possibility.
Thank you.
That would good question.
In all honesty, you're asking the dermatologist who works on that.
Absolutely, HPV specialist. Yeah, yes.
I think you never know. I think that as we understand the pathophysiology of what's going on, what's the mechanism of SMA, hopefully we'll be able to learn other avenues. I think it may take multiple different types of therapies that have a cumulative effect to give us long-term beneficial treatments. So all of these things may play into the ultimate role. And I'd like to emphasize what happens in a mouse, as Dr. Porter well knows, may not happen in a human. And so there's a lot more work to be done.
Now, we heard from you at the meeting that this process called autophagy, which is a rather newly identified, may well have something to do with SMA pathogenesis.
Although when it goes to human, you just don't know. You might find things in mouse, and that's been shown with other neurodegenerative diseases. What works in a mouse sometimes doesn't work in a human. Right.
Sylvia writes with an oft-told complaint. Many doctors have limited knowledge or no knowledge of SMA. Charlotte, this might be heading your way. It seems many times as parents, grandparents, we are trying to educate medical personnel about SMA. Many times our actions or inactions can even cause harm to our children. We as parents do much research on treatment, nutrition, and are knowledgeable of issues. What is your best advice about working with hospital specialists, insurance companies that don't listen to parents or that don't go by the SMA protocols? This is all new territory for so many of us. We cram so much information into our brains about SMA. It's aggravating when medical professionals disregard what we know to be true. We don't often have much choice in doctors because of the location of where we live. Your thoughts on this issue would be appreciated.
Yeah, I mean, we've touched on this a little bit already, and I think Dr. Porter made a good point about this. This is also an area where advocacy groups like FightSMA play such an important role, and it's really important to utilize them for help in this regard. Even if you're able to travel only once a year, for example, to see someone a little further away who's a real bona fide neuromuscular or SMA expert, that person can provide a lot of advice to local physicians. So one of the models that often works for patients that I take care of is to have a local neurologist who may be less familiar with the disease and travel to see an expert maybe once a year or every other year, and that person can act sort of more of a consultant, but can help really guide the more local physicians. And FightSMA and other groups can help find those experts that are closest to you, even if they might be a little bit of a distance. So that's one possibility to consider.
We're asked by Shannon about the use of TENS, capital T-E-N-S, or other electrostimulation on a type 1 15-month-old. Charlotte, I'm afraid this bounces back to you again. Electric stimulation in a type 1 baby.
So I'm not aware that that's something that's commonly suggested necessarily. I think there may be some neurologists who might feel that it would be indicated, but in general, I don't think that we believe that it's something that is a routine benefit necessarily.
Yeah. I think
Do you think that's fair?
Yeah, I think the evidence base for that does not exist. In our earlier remarks about first do no harm,
Right.
probably not, doesn't sound like possibly a great idea. There's someone asking about Celebrex, and I can just off camera just say this is a study that came from my lab. Low dose celecoxib or Celebrex has been shown in mice to have a modest impact, and so we believe that there may be an effect at a surprisingly low dose, but we have not yet proven that. There's a trial that's being planned to start within the next 6 months, and we hope by this time next year we may have an answer. If there is an impact of Celebrex, my sense is it will be a modest impact. It will not be to the same extent as one anticipates for gene therapy, ISIS, or the PTC compound. But anyway, it's something we hope to get some clarity on. There's a question again about trials in Europe from somebody in Switzerland. Chris, what was your understanding about what European trials? The Trophos trial was taking place through the EU?
I was just looking that up, but they just concluded the phase 2-3 trial, and so Trophos is waiting to move forward. And the Sanofi PTC collaboration has been ongoing. It is not open to the best of my knowledge. I'm sorry. Roche PTC Therapeutics has a phase 1 that is ongoing and is not open, but you should check with clinicaltrials.gov.
Okay. A question of whether a combination of ISIS and gene therapy is something that one should anticipate or would be worthwhile looking at from a therapeutic point of view. Brian, and then Adrian.
So with both of these therapies, be it ISIS or be it the gene therapeutics, these are experimental therapeutics. And how the regulatory structure currently is aligned is that any individual therapy has to be tested individually for its safety and for its then potential efficacy. One thing that I think we can appreciate in any therapeutic development on complex diseases like spinal muscular atrophy is that there's likely not a single silver bullet,
right,
gold bullet that is going to be the absolute cure. We certainly, in any of these therapies, aim for the grand slam,
Right.
And we hope for that. But at the same time, one has to realize that we have a number of therapeutics moving forward. That is the ISIS oligonucleotides. We've got the gene therapy. We can't plan exactly for miracles, and I think we need to be in an understanding in the clinical trial and the research stage to potentially combine therapies in the future. But I'm going to go, to say that ISIS will now want gene therapy added to their trial or vice versa. Let's see how things play out in clinical trials. But let me just tell you one aspect that one could imagine for a therapy. So you think about gene therapy and you think about ISIS, they're hitting the same target. They're upregulating SMN. So could there be an advantage to combining those? Maybe. We'll see. But you could think about now another sort of combination, and that is there's muscle weakness and we need to increase SMN in the motor neurons to preserve function. We may think about combining sorts of drugs and therapies for muscle enhancement as augmentive therapies and or physical therapies that could augment any of these sorts of therapies as adjunctive or secondary therapies. I think that we have to take this one step at a time, though, and not to say that we have the kitchen sink that is going to fix every single problem in SMA.
Right.
We have to run safe trials, we have to run efficacious trials, and then we can really think about combinatorial sorts of trials.
Great.
Yeah, I agree. I mean, so each of these things has to be proven in terms of safety and efficacy. But down the line, one can imagine that scenarios in which combination drugs could be effective. So, for example, something that's neuroprotective versus something that increases SMN levels. Or even if it hits the same target, but it turns out that one drug is more effective in the CNS and another drug is more effective in the periphery, and somehow the combination turns out to be more potent. That would have to be tested eventually. That's not where you would start.
There's a question while we have you, Adrian. When will ISIS be announcing phase 3 clinical trial site locations and start dates?
I can't really comment on that, but you should check clinicaltrials.gov or even the ISIS website, because they keep that pretty up to date.
Yeah. And so when I asked Frank Bennett, who was here at the meeting but couldn't stay, he said within the next 2 months. It was a trick question. I knew the answer.
Yes, but are you allowed to say?
No. He said, no, just keep watching. Within the next 2 months, they'll be making announcements. And we're talking for the 2 phase 3s enrollment of over 100 study entrants each. So there'll be a broad availability. Here, we're back in the uncomfortable realm of diets again, Charlotte. I apologize. Stephanie V. has written, my son started carnitine in 2011. He's now 16. He has SMA type 2. He started levocarnitor in 2011. He was quite super chunky, 5 feet tall. After he also started after the amino acid, after he started this, his weight plummeted to 60 pounds at age 16, and now he's 5'5", and still this weight has been kept off. So, is there a correlation with that and weight loss, to your knowledge?
With Carnitor?
Yeah.
I'm not aware of that correlation. The main data that we have regarding Carnitor and L-carnitine is from the valproic acid studies. We knew that valproic acid could actually cause carnitine deficiency, so they were given together.
Right.
And I'm not aware of any data that Carnitor or L-carnitine can cause, that is known to cause weight loss in and of itself.
Okay. And nor was I.
So, Ellis,
yes,
can I
John.
bring something up?
Absolutely.
It becomes very obvious to me as you're directing a lot of questions toward Dr. Sumner.
Yes.
That there's a major issue that a parent may not ask about. So I thought I'd volunteer
Good.
this.
You can see how it's struggling.
Frankly, there's not enough Dr. Sumners. So there's
we're
not enough.
We're going to clone Charlotte.
Yeah. There's not enough clinical researchers and practitioners in the neuromuscular field to help take care of children with SMA and other neuromuscular diseases. And so a lot of times parents ask, how can they make a difference? Well, one way you can really make a difference is that those of you that see physicians at academic medical centers, you're going to encounter medical students. You're going to encounter residents. You're going to encounter fellows. And, you can make a difference for what career path they choose. And so, if you encourage them to take the path toward becoming a Dr. Sumner, and then we're going to end up with more critical mass in terms of trained physicians in this field that can really help manage SMA. And that's a way you can make a huge difference. And I think just talk with the medical student, talk with the resident, and say, hey, are you interested in going into this? You can really help my child and other children. And I think that's a big way they can make a huge difference. So
That's an excellent point, John. We certainly, at the Fight SMA, and we're trying with our Emerging Young Investigator Awards, which we heard from 3 new ones this year, from the research point of view, but absolutely right, as far as bringing in more Charlottes. We all need more Charlottes, I think.
Be careful what you wish for.
To quote Oscar Wilde. Okay, so actually, we are coming close to the end of the questions. If there are any others out there, we've got another 10 minutes or so to go. And I do apologize for the ones we haven't touched on. They're really beyond our expertise, but we will be trying to get back to you with the which orthopedic brace to wear type questions. Brian, is there a way to make sufficient quantities of virus to adequately treat the SMA population once it passes FDA requirements?
That's an excellent question, and it's something that we've taken quite seriously, and we have emerged from an academic, we're running the first-in-human clinical trials as an academically run clinical first-in-human trial. But we have been fortunate to have garnered outside interest, and that is biotechnology companies and outside expertise. A new biotechnology company is formulated around our research, and the name of that company is Avexis Biotechnology. And one of Avexis's goals is to actually start working with key leaders within the AAV field, with ourselves in my laboratory, on thinking about scalability. And one of the things that certainly concerns any therapeutic development is that if you do have a drug, are we going to be able to make it in sufficient quantities? These are things that we have seen within the field, and problems within manufacturing to be able to make enough critical products, and we've seen this in the lysosomal storage diseases not too long ago on just manufacturing issues. This is something that I take personally extremely at a level of high importance, on the aspect that we have very strong preclinical data that would suggest that there's a lot of things that have to happen right in the human clinical trials. But if we're right, we have a potential to make a very large impact in spinal muscular atrophy. And I'm putting it not only on my shoulders, but now this biotechnology company's shoulders, Avexis, which has an outstanding group of people who I have personally been able to help handpick with true expertise to be able to go out and start working with other companies to be able to think about the scalability of this biological product. We've got a lot of people in the gene therapy field that is working together, and there's a large number of companies and academic groups that are really putting their heads together to try to overcome this challenge, because right now in the gene therapeutics, Chris said earlier, and he is certainly correct, gene therapy was in the graveyard. Things are changing, and there's a different tune and a different air in the gene therapeutic space. We're starting to see some clinical trials that are showing, in early clinical trials, but are starting to show signs of success. And this is Leber's congenital atrophy. We have patients who have never seen before, and now we have mothers who, for the first time in their life, they can see the color of their children's eyes. There's children now that have been treated a one-time gene therapy in a blindness disorder that can now navigate complex mazes for the first time. So I think there's a lot of promise within the field of gene therapeutics. A lot of people are paying attention to this. We don't have all of the answers today. We're exploring currently. Just Avexis has gotten into our hands a very scalable method of producing AAV, and we have to put the microscope down and really determine, is this a methodology that is going to be scalable for this if it works. I hope that we can be in the situation that we say we need to make a lot of this. And I think that we will, scientists and the production specialists will be coming in to really get behind many of these biotechnology companies to figure out to figure this out.
A very tricky one as our penultimate or antepenultimate question. Are we any closer to identifying the window for therapies for type 1, type 2, type 3? Oh, sorry about that.
I apologize. It's clearly time almost to wrap up. Are we any closer to identifying the window of therapy for type 1, type 2, type 3? Brian, are you feeling brave?
You know, this is an excellent question, and I think is going to be answered in human clinical trials. But I will talk about one of my friends, collaborators, and colleagues, Dr. Arthur Burgess, who we have started, and now with support from NINDS, we are generating pig models of spinal muscular atrophy to try to answer this question, and that is, can we develop larger models of type 1, type 2, and type 3, and really looking at different stages of therapeutic intervention and determining where we sit on therapeutic success. I think what we heard from this meeting, Christine DiDonato's group and others, Kat Lutz's group, is also producing mouse models of less severe animals where we can intervene at various time points for therapeutic intervention. What I will share, and it came out of this meeting, and this is really work that's run by Dr. Burgess's laboratory. We play some part in this, but what Arthur's laboratory has done in the pig model of SMA is treat a pig once it's gotten the initial signs of weakness. And I think there's very encouraging results that says that one can either stop progression of the disease or stabilize the disease and potentially have that pig do a little better than it did before its symptoms started to show. I think there's a lot of work to be done here, but there's great hope based upon some of these trials in the mouse studies and in now these pig studies that are starting to emerge to say that SMN upregulation is a viable and is likely a viable therapeutic target for type 1s, 2, and 3. Mm-hmm. Ultimately, though, we cannot make new motor neurons if motor neurons are lost. So I think we have to keep that into perspective.
Charlotte, thank you very much, Brian.
So I think one thing to think about is that these drugs may work in several different kinds of ways. So there is emerging data that in severe SMA, some of the motor neurons never quite form normally, that they're there, but they're sort of immature. So one way that drugs might work, particularly in the most severe individuals, is to help that maturation process along. Another way that they might work is to prevent motor neurons from being lost later. That is, a motor neuron that developed, preventing it dying. And then a final way that they might work, or maybe there's more than that, but another way that they may work is that it may allow motor neurons that are there to actually be even more robust in the sense that we know motor neurons have some capacity to sprout distally, so they can take over the territory of muscle fibers that have been denervated by motor neurons that have been lost, and therefore result in greater muscle strength because of this distal sprouting ability. So there are multiple ways, I think, that replacing SMN may benefit motor neurons. And it may be that in certain stages of the disease, they would work predominantly in one way. And at later stages in the disease, they might work predominantly in another way. But while the preclinical data would suggest the earlier the better, and that may be true as well in humans, I also am a believer that even later treatment is going to have some benefit.
Well, thanks. That's a very thorough, encouraging
Yeah, and
answer.
I think that's been shown in several preclinical models,
Right.
published and unpublished. You know, Brian was talking about the pig.
Right.
But even Dr. Kaspar's earlier work with the gene therapy product, where he delivered it at 2, 5, and 10 days, showed efficacy later on.
Right.
Arthur published work with ASOs in a severe model demonstrating a partial rescue, and totally shameless plug. Today, our lab published something with looking at day 1, 2, 3, 4, 5, 6, 7, 8.
Right.
Even at the last time point that we looked at, there was a therapeutic effect, and this is after symptoms have really kicked into a very severe model.
Right. And
To wrap it up with this aspect, I think what it says is that replacing SMN is always a good idea, and that's obviously a wonderful target.
Sure.
So, like Dr. Sumner was saying, it might depend upon what targets are available, what type of developmental stage you're addressing this disease at, that you'll get a different kind of readout or outcome for restoring SMN expression.
And I guess the PTC low-dose, high-dose thing also showed some recovery as well. There's a question about pigs as pigs, but Arthur has his pig, Dr. Larson, there were some rumors of SMA pigs emerging from Columbus, Missouri. What's their status?
So it seems to be the Columbus-Columbia pig duel.
Yes.
Porcine
cage match.
Exactly. And so the Columbia pigs have been under development, and they are transgenic. They are not quite SMA. It's a slightly different take on this scenario, where this will be a transgenic model similar to the SMA mice. And so we will be able to establish a colony that is SMN2 based, so knock out the pig SMN and have the human SMN2 gene. And there have been some hiccups along the way, but I think things are looking up at this point.
Okay, thanks. Charlotte, a quick question from Europe about salbutamol and its use in SMA. Do you put any of your patients on salbutamol?
I don't. So I don't think that we have strong clinical trial evidence at this point that it's efficacious. My own practice is not to prescribe drugs that have not been demonstrated to have clinical benefit in rigorous clinical trials.
Right. So maybe in the class
But
of...
I do know that a lot of individuals with SMA are taking salbutamol.
Right. And it may have an effect, but if it has an effect, it would be somewhat unlikely to have a profound effect. Yeah. I think we're close to being done. I don't know if there's any final words from any of the panel. Personally, I just am at the risk of sounding Pollyannish. It's never been as promising when we saw the evidence from ISIS, from gene therapy, the PTC compound. In addition, solid basic stuff from Elliott's lab looking at potential new avenues to take, and Chris's work as well showing the ASO further evidence. And then finally some of Charlotte's work showing some absolutely new neuroanatomy biology that Tom Crawford, and Kathy have been involved in. It was really impressive work. So on a variety of fronts, and having John there pushing from the sides and pulling it along, it really is a very positive sociology. It was a very good meeting, and I think the future is very bright. Thanks very much for joining us in this panel.
Thank you.
Thank you.
You can collect your honorarium from Danielle.
Really clear.